Unit 5, Human Physiology. The reductionist approach to study of life forms resulted in increasing use of physiochemical concepts and techniques. Majority of these study employed either surviving tissue model or straight away cell-free system. An explosion of knowledge resulted in molecular biology. Molecular physiology became almost synonymous with biochemistry and biophysics. However, it is now being increasingly realized that neither a purely organismic approach nor a purely reductionist molecular approach would reveal the truth about biological process or living phenomena. Systems biology make us believe that all living phenomena are emergent properties due to interaction among components of the system under study. Regulatory network of molecules, supramolecular assemblies, cells, tissue, organisms, and indeed populations and communities each create emergent properties. In the chapter under these unit major human physiological process like digestion, exchange of gases, blood circulation, locomotion, and movement are described in cellular and molecular terms. The last two chapters point to the coordination and regulation of body events at the organismic level. Scientist Alfonso Corti, Italian anatomist, was born in 1822. Corti began his scientific career studying the cardiovascular system of reptiles. Later, he turned his attention to the mammalian auditory system. In 1851, he published a paper describing a structure located on the basilar membrane of the cochlea containing hair cell that convert sound vibration into nerve impulse. The organ of Corti. He died in the year 1888. Chapter 16, Digestion and Absorption. Food is one of the basic requirements of all living organism. The major component of our food are carbohydrate, proteins and fats. Vitamins and minerals are also required in small quantities. Food provide energy and organic material for growth and repair of tissues. The water we take in plays an important role in metabolic process and also prevent dehydration of the body. Biomacromolecules in food cannot be utilized by our body in their original form. They have to be broken down and converted into simple substance in the digestive system. This process of conversion of complex food substance to simple absorbable forms is called digestion and is carried out by our digestive system by mechanical and biochemical method. General organization of the human digestive system is shown in figure 16.1. 16.1 Digestive system. The human digestive system consists of the alimentary canal and the associated glands. 16.11 Alimentary canal. The alimentary canal begins with an anterior opening, the mouth, and it opens out and posteriorly through anus. The mouth leads to the buccal cavity or oral cavity. The oral cavity has a number of teeth and a muscular tongue. Each tooth is embedded in a socket of jawbone that is given in figure 16.2. This type of attachment is called thecodont. Majority of mammals, including human being, forms two set of teeth during their life. A set of temporary milk or deciduous teeth replaced by a set of permanent or adult teeth. This type of dentition is called diphodont. An adult human has 32 permanent teeth, which are of four different types: heterodont dentition, namely incisor, canine, premolar, and molar. Arrangement of teeth in each half of the upper and lower jaw in the order eyes, incisor, canine, premolar, and molar is represented by a dental formula, which in human is 2123. The hard chewing surface of the teeth, made up of enamel, helps in the mastication of food. The tongue is freely movable muscular organ attached to the floor of the oral cavity by the frenulum. The upper surface of the tongue has a small projection called papillae, some of which bear taste buds. The oral cavity leads into a short pharynx, which serves as a common passage for food and air. The esophagus and the trachea open into the pharynx. A cartilaginous flap and epiglottis prevent the entry of food into the glottis. Opening of the windpipe during swallowing. The esophagus is a thin, long tube which extends posteriorly, passing through the neck, thorax, and diaphragm, and leads to J-shaped bag like structure called stomach. A muscular sphincter, gastroesophageal, regulate the opening of esophagus into the stomach. The stomach, located in the upper left portion of the abdominal cavity, has four major parts: a cardiac portion into which the esophagus opens, a fundic region body, main central region, and a pyloric portion which opens into the first part of a small intestine. A small intestine is distinguishable into three regions: a C-shaped duodenum, a long coiled middle portion jejunum, and a highly coiled ileum. The opening of the stomach into the duodenum is guarded by the pyloric sphincter. Ileum opens into the large intestine. It consists of cecum, colon, and rectum. Cecum is a small blind sac which hosts some symbiotic microorganism, a narrow finger-like tubular projection. The vermiform appendix, which is a vestigial organ, arises from the cecum. The cecum opens into the colon. The colon is divided into four parts: an ascending, a transverse, a descending part, and a sigmoid colon. The descending part opens into the rectum, which opens out through the anus. The wall of alimentary canal from esophagus to rectum possesses four layers that is given in figure 16.4, namely serosa, muscularis, submucosa, and mucosa. Serosa is the outermost layer and is made up of thin mesothelium, epithelium of visceral organ with some connective tissue. Muscularis is formed by smooth muscles, usually arranged into an inner circular and an outer longitudinal layer. An oblique muscles layer may be present in some region. The submucosal layer is formed of loose connective tissue containing nerves, blood, and lymph vessels. In duodenum, glands are present in submucosa, the innermost layer lining the lumen of the alimentary canal. In the mucosa, this layer forms irregular folds, ragi in the stomach, and small finger-like folding called villi in the small intestine. The cells lining of the villi produce numerous microscopic projection called microvilli, giving a brush border appearance. These modifications increase the surface area enormously. Villi are supplied with a network of capillaries and a large lymph vessels called the lacteal. Mucosal epithelium has goblet cells which secrete mucus that help in lubrication. Mucosa also form glands in the stomach that is called gastric glands, and crypts in the between the bases of villi in the intestine, crypts of liver cone. All the four layers show modification in different parts of the alimentary canal. 16.12 Digestive gland. The digestive gland associated with the alimentary canal include the salivary gland, the liver, and the pancreas. Saliva is mainly produced by three pairs of salivary glands: the parotid cheeks, the submaxillary, submandibular, lower jaw, and the sublinguals below the tongue. These glands, situated just outside the buccal cavity, secrete salivary juice into the buccal cavity. Liver is the largest gland of the body weight about 1.2 to 1.5 kg, and in an adult human it is situated in the abdominal cavity just below the diaphragm and has two lobes. The hepatic lobules are the structural and functional unit of liver, containing hepatic cells arranged in the form of cord. Each lobule is covered by a thin connective tissue set called the capsule. The bile secreted by the hepatic cells passes through the hepatic ducts and is stored and concentrated in a thin muscular sac called the gallbladder. The duct of gallbladder, that is cystic duct, along with the hepatic duct from the liver, forms the common bile duct that is given in figure 16.6. The bile duct and the pancreatic duct open together into the duodenum as the common hepatopancreatic duct, which is guarded by a sphincter called the sphincter of Oddi. Sphincter of Oddi. The pancreas is a compound, both exocrine and endocrine, elongated organ situated between the limbs of the C-shaped duodenum. The exocrine portion secrete an alkaline pancreatic juice containing enzymes, and the endocrine portion secrete hormones, insulin, and glucagon. 16.2 Digestion of food. The process of digestion is accomplished by mechanical and chemical process. The buccal cavity performs two major functions: mastication of food mastication of food and facilitation of swallowing. The teeth and the tongue, with the help of saliva, masticate and mix up the food thoroughly. Mucus in saliva helps in lubricating and adhering the masticated food particles into a bolus. The bolus is then conveyed into the pharynx and then into the esophagus by swelling or deglutition. The bolus further passes down through the esophagus by successive waves of muscular contraction. muscular co- Successive waves of muscular contraction called the peristalsis. The gastroesophageal sphincter controls the passage of food into the stomach. The saliva secreted into the oral cavity contains electrolytes, that is sodium, potassium, chlorine, and bicarbonate, and enzymes, salivary amylase, and lysozyme. The chemical process of digestion is initiated in the oral cavity by the hydrolytic action of the carbohydrate splitting enzymes. The salivary amylase, about 30% of starch, is hydrolyzed here by this enzyme. That's maximum optimum pH is 6.8 into a disaccharide into maltose. Lysozyme present in the saliva act as an antibacterial agent that prevent infection. The mucosa of stomach has gastric glands. Glands have three major type of cells, namely: first, mucous neck cell, which secrete mucus; peptic or chief cell, which secrete the proenzyme pepsinogen; and the parietal or oxyntic cells, which secrete HCl and intrinsic factor essential for absorption of vitamin B12. The stomach stores the food for 4 to 5 hours. The food mixes thoroughly with the acidic gastric juice of the stomach by the churning movement of its muscular wall and is called chyme. The proenzyme pepsinogen on exposure to hydrochloric acid gets converted into the active enzyme pepsin. The proteolytic enzyme of the stomach, pepsin, convert protein into proteoses and peptides. The mucus and bicarbonate present in the gastric juice play an important role in lubrication and protection of the mucosal epithelium from exfoliation by the highly concentrated hydrochloric acid. HCl provide the acidic pH, that is 1.8, optimal for pepsins. Rennin is a proteolytic enzyme found in gastric juice of infants, which helps in the digestion of milk protein. Small amount of lipases are also secreted by gastric gland. Various type of movements are generated by the muscularis layer of the small intestine. These movements help in a thorough mixing of the food with the various secretion in the intestine and thereby facilitate digestion. The bile, pancreatic juice and the intestinal juice are the secretion released into the small intestine. Pancreatic juice and bile are released through the hepatopancreatic duct. The pancreatic juice contains inactive enzymes trypsinogen, chymotrypsinogen, procarboxypeptidase, amylase, lipase, and nuclease. Trypsinogen is activated by an enzyme enterokinase secreted by the intestinal mucosa into active trypsin, which in turn activate the other enzymes in the pancreatic juice. The bile released into the duodenum contain bile pigment, that is bilirubin and biliverdin, bile salts, cholesterol and phospholipids, but no enzymes. Bile helps in emulsification of fat, that is breaking down of the fats into very small micelles. Micelles. Bile also activate lipases. The intestinal mucosal epithelium has goblet cells which secrete mucus. The secretion of the breast border cells of the mucosa, along with the secretion of the goblet cells, constitute the intestinal juices or succus entericus. These juices contain a variety of enzymes like disaccharides, example maltase, dipeptidases, lipase, nucleosidase, etc. The mucus along with the bicarbonate from the pancreas protect the intestinal mucosa from acid as well as provide an alkaline pH, that is 7.8, for enzymatic activities. Some mucosal gland, that is Brunner's gland, also helps in these proteins, proteoses and peptones, partially hydrolyzed proteins in the chyme reaching the intestine are acted upon the proteolytic enzymes of pancreatic juice as given below. Carbohydrate in the chyme are hydrolyzed by pancreatic amylase into disaccharides. Fats are broken down by lipase with the help of bile into di and monoglycerides. Nucleases in the pancreatic juice act on the nucleic acid to form nucleotides and nucleosides. The enzymes in the succus entericus act on the end product of the above reaction to form the respective simple absorbable forms. These final steps in digestion occur very close to the mucosal epithelium cells of the intestine. The breakdown of biomacromolecules mentioned above occurs in the duodenum region of the small intestine. The simple substances thus formed are absorbed in the jejunum and ileum region of the small intestine. The undigested, unabsorbed substances are passed on to the large intestine. No significant digestive activity occur in the large intestine. The function of large intestine are: first, absorption of some water minerals and certain drugs. Second, secretion of mucus which helps in adhering the waste undigested particles to get together and lubricating it for an easy passes. Okay. The undigested, unabsorbed substance called fecus enters into the cecum of the large intestine through ileocecal wall, which prevent the backflow of the fecal matter. It is temporarily stored in the rectum till defecation. The activity of the gastrointestinal tract are under neural and hormonal control for proper coordination of different parts. The sight and smell and or the presence of food in the oral cavity can stimulate the secretion of saliva. Gastric and intestinal secretion are also similarly stimulated by the neurosignals. The muscular activity of different parts of the alimentary canal can also be moderated by neural mechanism, both local and through the CNS. Central nervous system. Hormonal control of the secretion of digestive juices is carried out by local hormones produced by the gastric and intestinal mucosa. Calo- Calorific value of protein, carbohydrate, and fat. Boxed item not for evaluation. The energy requirement of animals and the energy content of food are expressed in terms of measure of heat energy, because heat is the ultimate form of all energy. This is often measured to as calorie or joule, which is the amount of heat energy required to raise the temperature 1 gram of water by 1 degree Celsius. Since this value of tiny amount of energy, physiologists commonly use kilocalorie or kilojoule. 1 kilocalorie is the amount of energy required to raise the temperature of 1 kg of water by 1 degree Celsius. Nutritionists traditionally refer to kilocalorie as the calorie or joule, always capitalized. The amount of heat liberated from the complete combustion of 1 gram of food in a bomb calorimeter, a closed metal chamber filled with O2. It is its gross calorific or gross energy value. The actual amount of energy combustion of 1 gram of food in the physiological value of food. Gross calorific value of carbohydrate, proteins, and fats are 4.1 kilocalorie per gram, 5.65 sorry 5.65 kilocalorie per gram, and 9.45 kilocalorie per gram respectively, whereas the physiologic value are 4 kilocalorie per gram, 4 kilocalorie per gram, and 9 kilocalorie per gram respectively. 16.3 absorption of digested product. Absorption is the process by which the end product of digestion passes through the intestinal mucosa into the blood or lymph. It is carried out by the passive, active, or facilitated transport mechanism. A small amount of monosaccharides like glucose, amino acids, and some electrolytes like chloride ions are generally absorbed by simple diffusion. The passes of these substances into the blood depend upon the concentration gradient. However, some substances like glucose and amino acids are absorbed with the help of carrier protein. This mechanism is called the facilitated transport. Transport of water depend upon the osmotic gradient. Active transport occurs against the concentration gradient and hence require energy. Various nutrients like amino acids, monosaccharides like glucose, electrolytes like Na plus are absorbed into the blood by this mechanism. Fatty acids and glycerol, being insoluble, cannot be absorbed into the blood. They are first incorporated into small droplets called micelles, which move into the intestinal mucosa. They are reformed into very small protein coated fat globules called the chylomicrons, which are transported into the lymph vessels, lacteals in the villi. These lymph vessels ultimately release the absorbed substance into the bloodstream. Absorption of substance takes place in different parts of the alimentary canal, like mouth, stomach, small intestine, and large intestine. However, maximum absorption occurs in the small intestine. A summary of absorption, site of absorption, and substance absorbed is given in table 16.1. The absorbed substance finally reach the tissue, which utilize them for their activity. This process is called assimilation. The digestive waste is solidified into coherent feces in the rectum, initiate a neural reflex causing an urge and or desire for its removal. The ejection of feces to the outside through the anal opening defecation is a voluntary process and is carried out by mass Peristaltic movement. 16.4 Disorder or digestive system. The inflammation of the intestinal tract is the most common element due to bacterial or viral infections. The infections are also caused by the parasite of the intestine like tapeworm, roundworm, threadworm, hookworm, pinworm, etc. Jaundice. The liver is affected. Skin and eyes turn yellow due to deposition of bile pigment. Vomiting. It is the ejection of stomach contents through the mouth. This reflex action is controlled by the vomit center in the medulla. Feeling of nausea. Proceed vomiting. Diarrhea. The abnormal frequency of bowel movement and increased liquidity of the fecal discharge is known as diarrhea. It reduces the absorption of food. Constipation. In constipation, the feces are retained with the colon as the bowel movement occur irregularly. Indigestion. In this condition, the food is not properly digested, leading to a feeling of fullness. The cause of indigestion are inadequate enzyme secretion, anxiety, food poisoning, overeating, and spicy food. Summary. The digestive system of humans consists of an alimentary canal and the associated digestive gland. The alimentary canal consists of the mouth, buccal cavity, pharynx, esophagus, stomach, small intestine, large intestine, rectum, and the anus. The accessory digestive glands include the salivary glands, the liver with gallbladder, and the pancreas inside the mouth of the teeth. Masticate the food. The tongue tastes the food and manipulate it for the proper mastication by mixing with the saliva. Saliva contains a starch digestive enzyme, salivary amylase, that digests the starch and convert into maltose disaccharide. The food that passes into the pharynx and enters the esophagus in the form of bolus, which is further carried out through the esophagus by peristalsis into the stomach. In the stomach, mainly protein digestion takes place. Absorption of simple sugar, alcohol, and medicines also takes place in the stomach. The chyme food enters into the duodenum portion of the small intestine and is acted on by the pancreatic juice, bile, and finally by the enzymes in the succus entericus, so that the digestion of carbohydrate, protein, and fat is completed. The food then enters into the jejunum and ileum portion of the small intestine. Carbohydrates are digested and converted into monosaccharides like glucose. Proteins are finally broken down into amino acids. The fats are converted into fatty acid and glycerol. The digested end products are absorbed into the body through the epithelial lining of the intestinal villi. The undigested food enters into the cecum of the large intestine through ileocecal valve, which prevent the backflow of fecal matter. Most of the water is absorbed in the large intestine. The undigested food becomes semi-solid in nature and then enters into the rectum, anal, anal canal, and is finally egested out through the anus. Thank you friends for listening this audio, and please post your comment if you want any kind of improvement. So मिलते हैं नेक्स्ट वीडियो में, तब तक के लिए बाय, and just stay tuned with the biology critic and enjoy the knowledge.